Okay, good afternoon. So we're now finishing up with the topic on proteins and enzymes. So enzyme is an important protein. So it's actually a catalyst. So it's actually a catalyst. So as we know, catalysts are chemicals or substances which help speed up a chemical reaction but is not consumed. Okay, so in order to understand this, we have what you call the ingay. Pamute na lang muna. Mute all. Okay, so in order to understand this, so we have this what you call energy diagram. So in the x-axis, you have the progress of the reaction. Yan.
King, so ano na yun? Kami dito.
Hindi lang, alala na ako
Tanas.
nila.
Rinig na rinig yung ingay sa loob ng ano nyo. Okay. So again, so the progress of the reaction is in the x-axis. So yung from reactants to products, yun yung progress of the reaction. So from being a reactant to a product. So the y-axis is about the energy involved. Okay, so the amount of energy involved in creating the product. Okay, so like a catalyst, they increase the rate of reactions. So 10 to the 12 times faster. So this is how fast the reaction proceeds. So without these enzymes, the reactions in our the reactions in our body will not happen, because it will take like for example, hydrogen peroxide. There's a specific enzyme that will break down hydrogen peroxide. So if that will be 10 to the 12 times more slower than what the enzyme is doing, then we will die, because the amount of hydrogen peroxide in our blood is poisonous enough to kill us. Okay, so we have these enzymes which break down the hydrogen peroxide in our cells, so that our cells will not die. So that's how important enzymes is. So without enzymes, we'll just die. So they are not changed at the end of the reaction. That's why they're called catalysts, and they are made up of proteins. So what it does is if you look at this graph, so let's look at this red line first. So normally in a chemical reaction, it will take so much energy to create the product. So the amount of energy is called the energy of activation. Okay. So the red line shows the uncatalyzed reaction, or the one without enzymes. So it takes so much energy to create the products that we need. So what an enzyme does is it will help find another pathway na hindi ganun kahirap yung energy of reaction. Okay? So tingnan natin, for example, let's have this as an example. You have H2 plus I2. So it will form what you call an activated complex, when either you form the product or it goes back to the reactants. Okay? So pag nagsama sila, nagde-decide sila kung merong patutunguhan yung relationship nila. So ayan, mag-form sila ng products, HI. Or kung di kaya, babalik sila sa reactants. So the amount of energy needed to create this product is the energy of activation, or yung shortcut niyan EA, or energy of activation, EACT. That's just the energy of activation. Okay, so yun, kailangan yung mga enzymes na yan, because otherwise we cannot break down toxic chemicals in our body or make certain reactions to occur. So pati yung pag-unwind ng DNA natin, kailangan mga enzymes yan. Okay, so almost all of the processes in our body requires the use of enzymes. So most of enzymes are globular proteins, meaning they're water soluble. So although proteins are not the only biological catalyst, but for the most part, they're made up of proteins. So they are specific. Excuse me. Yes?
Sir, may pumapasok po. Hindi niyo pa po na-admit. Thank you po.
Sino? Wala ako
Sir,
nakita dito.
Wala.
Wala
Sige
dito
po.
sa room. Wala sa waiting. Wala akong nakita dito. Rejoin na lang.
Sige po, sasabihan ko po.
Ayan na. Medyo nakita ko na. Ano complete name nyo? Pakisulat yung complete name nyo, kasi when you join our class, dapat yung name na kilala ko. Kasi kung hindi ko kayo kilala, I can choose not to include you. Kasi baka may outsider, no? Okay, again, so most enzymes are specific. So may yung pangalan pa lang, no? Pag halimbawa yung naka-record ba to? I hope this is still recording, no? Okay. So for enzymes are specific. So ibig sabihin, pag may lactase enzyme ka, specific siya sa lactose. Atake niya yung lactose na molecule. And then you have chemicals like trypsin, which cleaves the peptide bonds of protein. So specific siya sa peptide bonds ng mga proteins. Wala siyang ibang target kundi yung peptide bonds ng proteins. And these enzymes are localized according to the need. So kung saan siya yung enzymes halimbawa na kailangan para ma-absorb ang nutrients natin nandun lang sa small intestines. Yung mga digestive enzymes natin nandun sa stomach. So, it's localized. Okay? So, hindi ka pwedeng for example, yung enzyme mo na may ano ka, for example, enzyme for a specific, halimbawa, lactase, no? Hindi ka pwedeng uminom ng lactase na enzyme. Kasi sa katawan pa lang, sa stomach pa lang, breakdown niya yung enzymes eh. So kung ano man yung kinakain mong enzyme, breakdown niya ng katawan to amino acids, and basically your body will create the enzymes needed. Okay? So yung may mga anong tawag diyan? May mga medical hoax or parang mga scam na nagbebenta ng enzymes. Meron ba? Meron kayong alam nun? Nagbebenta ng enzymes para daw lumakas ang katawan. Well, basically, meron kayong alam nun? Basically, your enzymes are formed here inside their body. Ano yung alam nyo?
Sir, yun ba yung mga sa frontliners na nagsasabing nagbo-boost ng ano niya po?
Iba naman yung vitamins, yung mga coenzymes, iba yun. Yung coenzymes yan, hindi naman nababreakdown yan. Yung enzymes lang na yung large kasi yan na hindi naman agad yan. May specific area lang siya sa body na nag-work. Okay? So, hindi automatic na may kakainin kang enzyme. Yung enzyme na yun aabot dun sa area na kailangan ng body. Okay? Basically, for the most part, yung mga enzymes na na digest natin, be-break down yun sa amino acids, and our bodies will form the enzymes that are needed. Okay? So, sa DNA natin, pag wala yung code for that specific enzyme, magkakaroon tayong mga genetic diseases. Like, for example, yung lactose intolerance. Yung lactose intolerance, absence yan ng the enzyme lactase na nagka-target sa lactose. Kaya sumasakit yung tiyan pag lactose intolerant kasi wala kang means para i-digest ang lactose. Okay? So yan, may kinalaman sa DNA yung kung may mutation sa DNA, may mga specific enzymes na hindi tayo nagagawa, it will have an effect on our body. Okay? Okay, so there are 6 types of enzymes. We can classify them into 6 types. We have the oxidoreductase. So this in this type of enzyme, okay, excuse me. So this type of enzyme helps the oxidation reduction reactions to proceed. So basically, this involves transfer of electrons. Pag may gain of electrons, it's reduction. Pag may loss of electrons, it's oxidation. So you have this type of enzymes, you have the oxidase, and you have the reductase. And then you have another type of enzyme. So this type of enzyme transfers a group between 2 compounds. So it's called the transferase. Okay, self-explanatory. It helps transfer a group, yung functional group or hydrocarbon chain between 2 carbons. Okay. And then you have hydrolase enzymes. So this breaks down basically yung nagkakaroon ka ng hydrolysis. So with water, you actually form you break down a molecule by adding water. So hydrolysis reactions. So pag breakdown natin yung mga digestive enzymes, most of it are hydrolases. And then you have lyases. This adds or remove groups involving a double bond without hydrolysis. So without the use of water molecules, you can add and remove groups, which involves a double bond. So isomerases, these rearrange atoms in a molecule to form an isomer. So basically rearrangement lang within the molecule. And finally, you have ligases, which form bonds between molecules. Okay. So basically, yung target ng enzymes is what you call a substrate. So like I mentioned kanina, yung lactase, it targets the lactose molecule, the lactose disaccharide, para break down into galactose and glucose. And then there are other enzymes which, like catalase, it targets hydrogen peroxide para break down into water para hindi mag-build up sa cells natin. So yung specific molecule na target ng enzyme is called the substrate. So these are the compounds whose reaction an enzyme catalyzes. So malaki yung enzyme, no? Di ba folded yan? Quaternary structure yan eh. Folded and folded again. And then there's a specific site lang dun sa enzyme na you call the active site. So this is the specific portion of the enzyme in which a substrate binds during a reaction. So for example, by breakdown niya yung ito for example is hydrogen peroxide. So specific yung site na yun for hydrogen peroxide, walang ibang molecule. So i-break down niya into to water and an ion. So basically, you have these products. Okay, so an enzyme catalyzes a reaction by attaching to a substrate at the active site. So yan, kakabit yung substrate dito sa active portion ng enzyme. So what you form here is what you call the enzyme substrate complex or the ES complex. Tapos eventually, magse-separate siya, you have the enzyme again, and then finally you have the products. So you form and release the products. So may mga theories yan kung paano nagpo-function ang enzymes. So the first model is called the lock and key model. So in this theory, yung enzyme mo is specific talaga dun sa shape ng substrate mo. Okay? So specific talaga siya sa shape ng substrate mo. So it will attach itself exactly as a lock, yung 'di ba pag may susi ka sa pinto, specific yung lock dun sa pinto. So rigid yung sa model na to, rigid yung enzyme. So an enzyme binds only substrate that exactly fit the active site. So the enzymes are analogous to a lock. And the substrate is the key that fits into the lock. Sa ibang model, ito yung mas accepted. You have the induced fit model. So medyo flexible yung enzyme. Tapos nag-a-adjust na yung enzyme pag nandun na yung substrate mo. So the enzyme and the substrate adjust the shape of the active site to bind to the substrate. So nag-increase yung range of substrate specificity. So hindi pwede sa ibang substrate, it could not induce the structural changes and no catalysis would occur. So ito yung induced fit model. Okay, so what are the factors? What are the factors affecting enzyme activity? So, the activity of the enzyme is how fast an enzyme catalyzes the reaction. So, number one factor is the temperature. Okay? So, pag sobrang lamig, hindi nag-function ng enzyme. Pag sobrang taas naman, nasisira yung protein structure niya. And then you have also the pH. Kaya importante yung buffers. May lecture ako dyan, no? Hindi ko, may lecture pala sa lab na i-include ko pala dun sa GC. So, mamaya na lang. Okay, and then you have substrate concentration. So, gaano karami yung concentration ng chemical target mo. And then you have also enzyme concentration. So, these are all positive effects kung for the substrate concentration, enzyme concentration, these are all positive effects. So, yung temperature and pH depende kung favorable sa enzymes. Itong enzyme inhibition, ito negative yung effect. So, it will try to inhibit. So, hindi mag-function yung enzyme, kaya enzyme inhibition. Okay, so let's talk about temperature. Okay, so for temperature, there's a specific range na nagko-function ng enzyme. Okay, so at low temperatures, enzymes show little activity. So it's not enough amount of energy for the catalyzed reaction. So pag low temperature yan, kaya tayo naglalagay sa ref, no? So para hindi masira yung mga pagkain natin, para yung mga bacteria na may enzymes, hindi maka-function yung enzymatic activity niya, so nag-slow down yung enzymatic activity, so di nasisira yung pagkain. And then, of course, at very high temperatures, you're actually destroying the protein structure. So the enzyme is destroyed, and so you have basically you denature the tertiary structure. Okay? So napapansin mo, anong tuwing kailan nasisira ang pagkain? Anong usually yung temperature yung mabilis masira?
Mainit po.
Medyo warm, no? Medyo summer. So, around 30 plus, madaling masira yung food. So, optimum temperature niyan, 37 degrees or our body temperature. So, at that temperature, madaling masira ang ano natin. Oops, matatapos na. So, at that temperature, madaling mag-function ng enzymes. So, at 30 to 37 degrees. Okay, pH naman. So, yung level of acidity and basicity. So, the optimum pH temperature in our body is 7.4. So, when you change that, when you lower or make your pH higher, then it will change the shape of the enzymes. Kasi nga, gawa sa amino acids, and amino acids will be affected by the change in pH. So, the optimum pH in the stomach is 2. So, yung mga enzymes mo na nasa stomach like pepsin, so it will function only in pH of 2. Okay? So, pag small changes in pH yan, some damages of the enzymes are reversible. So, pag bumalik siya sa same pH, babalik uli yung structure niya. Okay. So, when we talk about substrate concentration, so if you increase the enzyme concentration, the rate of reaction increases. Okay, so it makes sense naman, no? yung enzyme mo kasi, mas marami kang enzyme, mas marami kang pwedeng mga substrate molecules na i-transform into product. Okay? Pero kung pag-usapan natin yung substrate concentration, so kahit increase mo yung substrate concentration, there will be a point na magla-level off siya kasi limited naman yung enzymes mo. Okay? So mag-increase pa rin yung rate of reaction as you increase the substrate, pero eventually it will reach to a point na lahat ng enzymes mo naka-combine na. So wala nang ma-react na substrate. So the rate of reaction reaches its maximum where all of the enzymes are combined with. Please write down your complete name. Do not use a nickname. Okay? So eventually it will reach to a point na mag-level off siya. Okay, so what are these inhibitors or causes the enzyme to not function as well as could be expected? So these inhibitors cause enzymes to lose catalytic activity. So there are two types of inhibition. So there is a competitive inhibitor and a non-competitive inhibitor. So let's talk about the competitive inhibitor first. So, ayan. Example yan. Nakita ko ito sa internet eh. Sabi ko, okay. Mas maintindihan nila yung pero luma na tong TV show na to eh. Anyway, so ayun yung talagang target nyo, yung substrate. So ang competitive inhibitor, may nakiagaw. Okay, so ito dapat substrate yung kakabit sa active site, may ibang kabit. So yun, tama-tama sa sa image, no. So yun, may ibang kumabit. So dahil sa inhibitor na yan, hindi nag-function yung enzyme. So imbis na magkaroon ng product ka, yung substrate mo will remain as a substrate. So the inhibitor has a structure that is similar to the substrate, so it competes for the active site on the enzyme. So what you have is the solution there is to increase substrate concentration. So pag mas maraming substrate, mas maraming, so matatabunan ng inhibitor. So this will solve the problem by increasing the substrate concentration. Okay? So, yung competitive inhibitors, if it fits, it hits. So, nandyan lang. So, siya dapat yung makakabit. So, wala nangyari dito sa competitive inhibitor na walang actual na function, walang actual product yung mafo-form. Kumabit lang yung competitive inhibitor. So, wala lang makakabitan tong mismong target molecule natin. So remember that the inhibitor, okay, so now we talk about non-competitive. Okay, ano ba tong non-competitive? So inhibitor is not similar to the substrate and it does not compete for the active site. So hindi kaagaw ng inhibitor ng substrate mo. So ang nangyayari is it will bind to the enzyme and change the shape of the enzyme. So permanent to. Once na sinira niya yung structure ng enzyme, hindi na siya mag-function. Unlike yung dun sa competitive inhibitor, yung pag natanggal naman yung competition mo, magka-function yung enzyme. Ito, may sisira talaga dun sa enzyme mo. Okay, so yan, kaya nga masama yung heavy metal ions like lead, silver, mercury. So this will bond with the carboxylate groups and the hydroxyl groups of amino acids in an enzyme. So yun, merong bad effects dun sa enzymes. So kaya yung mga heavy metals na yan, permanent, mahirap kasi matanggal yun sa katawan. Unlike pag poison, pwede mong isuka. Yung mga heavy metal ions yan, mag-accumulate yan by binding with the enzymes that you have inside your cells. So, mahirap matanggal yan. Okay. So, one of the first antibiotics was penicillin. And how it works is it inhibits an enzyme needed for the formation of cell walls in the bacteria. So, yung penicillin yan, one of the first antibiotics. So, pinapatay niya yung bacteria by targeting the enzyme needed for the formation of cell walls. Pag wala yung cell walls, mas masisira yung bacteria and the infection is stopped. Okay? So, na-discover yung penicillin from a fungi in American pronunciation. So, penicillin basically came from a fungus. Okay, so what is the solution for this? So some chemical reagent should be able to remove the inhibitor. So kailangan matanggal yung mismong inhibitor na yan. Okay? So kung ang target mo is patayin yung bacteria, so pwede mo tong gamitin yung concept ng non-competitive inhibitors para masira yung bacteria. Pero kung target mo is for the health of the humans, ayaw mo magkaroon ng competitive inhibitor. Okay? So yung mga antibiotics, yung target niya is to make the bacteria not function. So minsan tina-target niya yung specific enzyme para masira yung organism. Okay, I think matatapos na to. So I'll create a new link for this. Okay, so just join, I think the same link will do. So please